This is the Chapter 9, Part 1 video. We're going to be looking at microbial genetics, following the blueprint of life from DNA to RNA to a protein. That flow of information of DNA to RNA to a protein is referred to as the central dogma of molecular biology. DNA is transcribed into RNA and then translated into a protein. The bacterial genome, the DNA, is the complete set of genetic information. So there's chromosome, and some bacteria have plasmids. So remember, the chromosome is single and circular in bacteria, and some may or may not have plasmids. The chromosome of E. coli, for example, is 4.7 times 10 to the 6 base pairs long. So a base pair is an A and a T, an adenine and a thymine is one base pair. So 4.7 million of those base pairs, A's and T's, C's and G's, in some sequence making up the E. coli chromosome. The size of an average bacterial gene is about 1,000 base pairs, and E. coli has about 3,500 genes. Just as a comparison, humans have about 30,000 genes. Plasmids, like we said before, they contain genes that are non-essential for normal cell function. They may give the cell new characteristics such as antibiotic resistance, toxin production, or virulence factors, as a few examples. The gene is the functional unit of the genome that codes for a product. Some genes are only transcribed into RNA, and RNA is the product. The majority of genes are actually translated into a protein. Looking at some characteristics of DNA, it's a deoxyribonucleic acid. It's composed of individual monomers referred to as nucleotides. It has a phosphate attached to the pentose sugar deoxyribose, and then it has the sugar, so we say it has a sugar phosphate backbone. Attached to each of the nucleotides is a nitrogenous base, an adenine, thymine, guanine, or cysteine. And those individual nucleotides are joined by covalent bonds between a 5' phosphate and a 3' hydroxyl. The 3' and 5' refer to the number carbon atoms on the pentose sugar. I'll show you that here in the drawing board. So here I've drawn a base pair, an adenine and a thymine. So with the nitrogenous base, they are attached to a sugar. Here's our pentose sugar. There's an oxygen here, and there's a different color here. At this beginning at this oxygen and going clockwise, there's a carbon here, here, and then the CH2. So it's a 5-carbon sugar, pentose sugar. It's a pentagon. And if we count those carbons, we can get the designation of the 5' and 3'. Beginning with the oxygen moving clockwise, we have 1, 2, 3, 4, and 5. So like I said in the previous slide, we have the 3' hydroxyl group, and we have the 5' phosphate group. So we have what we say is a 3' end and a 5' end of the nucleic acid. DNA is also antiparallel. The two strands of DNA, so DNA is double-stranded and complementary, so A's and T's attached to each other, G's and C's attached to each other, and they're oriented in opposite directions. So here you can see this is a nucleotide, basically what I drew on the drawing board. We have the oxygen group, the pentose sugar, 1, 2, 3, 4, 5, with the phosphate group. So we have the 5' end on this molecule, and then the 3' end down here at the hydroxyl group. The opposite strand, you can see the nucleotides are drawn upside down. We have the oxygen group, 1, 2, 3' hydroxyl, 4, 5' phosphate. So here are the 3' end, 5' end. So looking at this, we have the 5, 3, 3, 5. So they're oriented in opposite directions. This comes into importance when we talk about the process of DNA replication, because each of these strands are going to be replicated using the same enzymes but go in different directions. The base pairing characteristics, like I mentioned, adenine base pairs with 2 hydrogen bonds to thymine, guanine base pairs with hydrogen, guanine base pairs with 3 hydrogen bonds to cytosine. Characteristics of RNA, they're basically very similar with some exceptions. I have a deoxyribonucleotide here and a ribonucleotide down here at the bottom. Here you see the ribonucleotide. The only difference here is this extra hydroxyl group at the 2' position. Here we have no hydroxyl group at the 2' position for the DNA nucleotide. RNA is made up of the ribose sugar. So here we have the deoxyribose sugar, ribose sugar for the RNA molecule. RNA has uracil as the base instead of thymine, so we don't find thymine in RNA. RNA is usually single-stranded, it's usually linear, not helical, and it's usually found in shorter strands. There are 3 functional groups of RNA. There's messenger RNA, ribosomal RNA, and transfer RNA. Ribosomal RNA and transfer RNA are not translated into protein. They are the ultimate products. Each of those ribosomal RNA and transfer RNA are involved in the process of protein synthesis. Messenger RNA, which I don't have on this slide, is the temporary copy of the genetic code or DNA, and that messenger RNA is going to be translated into a protein. So we're going to look at some characteristics of bacterial replication before we get into transcription and translation. And before a cell divides, the DNA must be replicated, creating a second DNA molecule. So basically, DNA replication is needed for reproduction. Each cell after the division will have the same exact genetic information. So the offspring, quote-unquote offspring, are exact clones of each other. In these terms, offspring and parent cell, don't really apply to bacteria because both bacteria, after binary fission, are completely mature cells. You cannot designate which is the cell that started the process of replication and which is the new cell. They're both exact clones. So DNA replication occurs bidirectionally. So here, if you can imagine this picture here being a circular chromosome, double-stranded circular chromosome, we have an origin of replication where we get these replication bubbles. So we have this bubbling that occurs. At each end of the chromosome here, we have what we refer to as a replication fork. So DNA replication is going to occur to the right and to the left. This allows for the replication of the entire chromosome to be replicated in a shorter amount of time. DNA replication is also semi-conservative. Each of the 2 new molecules contain 1 original strand and 1 new strand. I'll demonstrate this on the drawing board as well. Alright, here I've drawn what looks like basically a railroad track, representing our original strand of DNA. So it's in red here. What's going to happen is the enzyme would break these railroad ties, making single-stranded DNA, so different enzymes can read the template DNA and make new strands. So after 1 round of replication, what we would have is 1 cell having an original strand, the other cell having an original strand, and then each having a new strand highlighted in blue. All right, so our 1 bacteria would have original and new, second bacteria original and new. Semi-conservative. Now, there's many different enzymes required to coordinate the process of DNA replication. The enzyme that we're going to talk about here first is DNA polymerase, and DNA polymerase is the powerhouse. It's going to be the workhorse of the process. It's going to be reading template DNA and adding nucleotides, synthesizing new strands of DNA. So it reads the template strand. So here in red, down here is the template strand. It's going to read this T, and it's going to incorporate an A into this new strand of DNA. The thing with DNA polymerase is it can only add to a 3' end. So it adds to the 3' end of the new strand, so the growing strand. So we say that new strand elongates in a 5' to 3' direction. DNA polymerase can only add to that 3' end because of the energy requirements. It's energetically easier to add to 3' end. If it added to the 5' end, it would take too much energy to actually do so. The process of DNA replication is very accurate. So one mistake might occur every 10 billion nucleotides. And DNA polymerase has a proofreading ability which allows it to correct some of the mistakes that it does make. So in one round of replication, a mutation, a mistake, is not likely to happen. If you remember back, E. coli has about 4.6 million base pairs. So roughly, we'll say 10 million nucleotides that would need to be replicated in a given round of cell, for a round of cell division. So in one round of replication, a mutation is not likely. A mistake is not likely. But if you consider how fast E. coli can grow, 20-minute generation time, and consider we can have 1 billion bacteria in 24 hours, given the space and nutrients for the bacteria, 1 billion times 10 million nucleotides, you can guarantee you have mutant cells in test tubes that we use in our lab. So what those mutations mean, we will talk about in the second video for Chapter 9. We're going to look at the process of replication, and we're going to break it into 6 or 7 steps, depending on how you take your notes. But our step 1 is specific proteins recognize the origin of replication. That allows helicase to attach to those proteins and begin the process of separating double-stranded DNA. So it's going to break the hydrogen bonds and form single-stranded template DNA. Primase is an enzyme that's going to synthesize small fragments of RNA called primers. The primers are the starting point for DNA polymerase to attach to. So DNA polymerase recognizes the primer and can attach to that. And the primer also provides a 3' end. As I mentioned, DNA polymerase needs that 3' end in order to make add nucleotides and make a new strand of DNA. So here we have an image with helicase. It's going to be going to the left, separating these two strands of DNA. Primase in purple here is going to synthesize these small RNA primers, providing a 3' end for DNA polymerase. Now, synthesis is continuously proceeds in on one strand in a 5' to 3' direction towards helicase. So, due to the directionality of DNA, since it's antiparallel, we're going to have these strands replicated a little bit differently, basically going in different directions. So the leading strand is going to be continuous in a 5 to 3 direction toward helicase. Now, like I have here in parentheses, steps 4 and 5 occur at the same time, so you could just add this to step 4 if you wanted to. The lagging strand, so the opposite strand of the leading, is going to be replicated a little bit differently. Since DNA polymerase cannot add to a 5' end, basically what happens is we have an interruption in the synthesis process. So synthesis has to be reinitiated periodically as helicase unwinds the DNA. And I'll show you a video of this here in a moment to give you a picture of this process as well. Since we have primase adding RNA primers, we need to get rid of those primers because DNA has DNA molecules in it, not RNA. So there's a different DNA polymerase that removes the RNA primers and adds DNA nucleotides. Now, in your book, the process is a little bit more complicated with the naming of the DNA polymerases. If you remember, DNA polymerase is going to be the enzyme doing steps 4 and 5. We're just going to say a different DNA polymerase is going to be the enzyme that's going to be removing the RNA primers. So steps 4 and 5, DNA polymerase. Step 6, a different DNA polymerase, just to simplify the process. Now, what happens when the step 6, we have that different DNA polymerase removing the RNA primers, what happens is they can't form covalent bonds where the DNA molecules are meeting. So DNA ligase seals the gaps, we say, forms a covalent bond where the RNA primers were removed. All right, so here is that expanded picture now. Here we still have helicase. We have the RNA primase putting down the RNA primer. You can see that in pink here. You see here that we have 1, 2, 3, 4 primers on this lagging strand. So the bottom one here is the lagging strand. DNA polymerase attaches to that primer and then adds nucleotides to the 3' end. It's going to stop when it reaches the other primer. So we get this leapfrogging that's going to occur as helicase separates DNA. This top strand is the leading strand because it's continuously synthesized toward helicase in a 5' to 3' direction. I'm going to show you a video now to give you a better picture of the process. There are many different videos that you can find on YouTube. I like this video just because it kind of simplifies things. It basically goes through the steps that we just talked about.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. DNA polymerase always begins a new strand by adding to an existing RNA primer, shown here in red, that has been constructed first. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, and DNA polymerase can only add nucleotides in one direction, the new strands must be made in different ways. One new strand, the leading strand, seen here in light blue at the top, is built continuously. The other new strand, the lagging strand, seen here in light blue at the bottom, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adenine always pairs with thymine. Guanine always pairs with cytosine. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A protein clamps the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA. However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
Alright, so hopefully that video helps you visualize that process of DNA replication. When you're taking your notes, like I said, we had 7 steps. Steps 4 and 5 were basically occurring at the same time. Now we're going to switch gears into gene expression. Gene expression basically can be defined as the production of a protein from a gene sequence. And that involves the two processes of transcription and translation. Transcription, or making RNA, what happens is DNA is used as a template, and most genes code for proteins. And we did say before that we do have RNA products as well. So messenger RNA is going to be transcribed in this process, and that messenger RNA is going to be translated then into a protein. And with transcription, we have an initiation process where RNA polymerase recognizes a nucleotide sequence on DNA called a promoter region. So RNA polymerase, different than DNA polymerase, is going to be the workhorse in transcription. Very important to be able to remember the distinction between RNA and DNA polymerase. Elongation occurs where RNA polymerase initiates synthesis without a primer, can do everything on its own basically. So it will separate the double-stranded DNA, attach, read, and synthesize all on its own. Then termination occurs when RNA polymerase encounters a terminator sequence. All right, here I have a template DNA strand. We know it's DNA because there's thymine in it. Okay, RNA polymerase would read this strand and synthesize RNA from this template strand. So where we would have an A, or where we have an A in DNA, we would have a U in messenger RNA. We have T to A, T to A, G, C to G, C to G, A to T, or sorry, oh, made that mistake. A to U, C to G, G to C, A to U, T to A, C to G, and G to C. So our DNA base pairing rules, if we're going DNA to DNA, if we have an A in the complementary strand, it's going to be a T. If we have a C, it's going to be a G. Okay. Same thing, if it's a T, it's going to be an A. If it's a G, it's going to be a C. But when we go from DNA to RNA, where we have an A in DNA, it's going to be a U in RNA. We have a T in DNA, it'll be an A, C to G, and G to C. Okay, so very similar to the DNA base pairing rules, but there is no thymine in RNA. So transcription, we're going to make messenger RNA. Translation is where we're going to take that messenger RNA using ribosomes and transfer RNAs and make a protein. So like I mentioned, that messenger RNA is the temporary copy, and the messenger RNA code is deciphered using the genetic code, which correlates sets of 3 nucleotides, referred to as a codon, with 1 amino acid. There are 64 codons, and 61 of them translate to 20 different amino acids. The other 3 codons are stop codons, so they stop the process of translation. So what this means is, since we only have 20 amino acids, but there's 61 codons that translate to an amino acid, is that there's more than one codon that can code for one specific amino acid. Here's a look at our genetic code, and you can see, like leucine, there are 6 codons. Here's a codon UUA, UUG, CUU. All of those would code for leucine. Serine, there's 4. Proline, there's 4. You can see the stop codons highlighted here. AUG is our start codon. It's going to start the process of translation, and it's also a methionine amino acid. So the ribosome is the site of protein synthesis, and that's where all this stuff, messenger RNA, the transfer RNAs, they put everything into a favorable position so we can catalyze a peptide bond. The transfer RNA are a sequence of RNA that carry the amino acids. They act as a key that interprets the genetic code. And that recognition is possible because they have an anticodon. So it's a set of 3 nucleotides at the one end of the transfer RNA that will read the codons. Here we have the transfer RNA molecule, RNA. We have the amino acid aspartate and the anticodon CUG reading the codon GAC. Okay. I'm going to go to the writing board again, and I'm going to give you some examples and show you how to use the genetic code. All right, here I have the genetic code up here. I have the DNA sequence. Take a moment to write that down. We're going to make this DNA, transcribe it into messenger RNA. So again, if we have an A, we'll have a U. All right, so now we have our messenger RNA. And the messenger RNA is the codon. So technically, UAA is a codon, UGG is a codon, UUA is a codon, AUU, and so on. So each 3 are a codon. But what we need to do is we need to look for the start sequence. We need to look for the first AUG. The first AUG is our start sequence. So our amino acid is going to start to be translated where we find that first AUG. Okay, looking at our AUG here, methionine, that's our start. So what we're going to do when we find that AUG, we're going to write start. The first AUG doesn't actually code for a methionine. So that means our next codon is going to be GUU. In order to use this genetic code, okay, looking at GUU, here we have our first letter in this column, UCAG. Since ours is G, we're going to look down here. So the first letter determines which box you were, or which row you were in. So we're in this row here. So that means our amino acid is going to be one of these. Looking at our next nucleotide, U, here's our middle letter, UCAG. This middle letter determines which column you're in. All right. But since the first letter designated that we're in this row, that means that we're going to be in this box. Okay. Third letter, U, looking over here, our last letter, looking down here, is a U. We follow that over, valine. Of course, as you can see, it doesn't matter what it would have been for that third letter, they all would have been valine. So we would write val. All right, A, A, follow that over, lysine. Of course, I call this genetic code, I call this the cheater code because it gives you the codon right next to it. The one in your course pack in your chapter 9 area, there's a genetic code that does not give you the codon right next to this amino acid. So you need to know how to use this genetic code as I highlighted before. All right, and then our next one is U, G, A. It's a stop codon. So you stop. So just because there's another one here doesn't mean that we continue. So we start with a start and end with a stop. Now in that worksheet, there is also a section for tRNA. So I can ask you, given a sequence of tRNA, and you can work backwards to get the messenger RNA. All right, so our tRNA, we're going to follow a base pairing rule from RNA to RNA. If we have a U, A, C, G, our coding units, we have a U, it's going to be an A. If we have an A, it's going to be a U, C to G, G to C. So our tRNA, all right, just going from mRNA to tRNA, U is A, U, this is a C, A, U, A, C, U, C, C, A. All right, so that would be our tRNA molecule. Now the thing is that remember the tRNA is the anticodon, and all this does is it reads the messenger RNA and it brings the amino acid. In order to use this genetic code here, you have to use the codon in this. One of the big mistakes people make is they will get a tRNA sequence, like I just showed you how to do that, and they'll use it in this genetic code, which is not correct. All right. The only reason why I have that in those practice worksheets is to show you that you can have a tRNA code and you can get messenger RNA. If you have messenger RNA, you can get a DNA sequence. That's all the purpose of that for those worksheets. All right, the last topic in this first video is regulation of gene expression. And regulation occurs due to changes in the bacterial's environment. So there's energy efficient strategies such as controlling the synthesis of the enzyme, so controlling the production, the making of the enzyme, and making only what is required so the cell isn't wasting energy. So why bacteria regulate gene expression is to conserve energy. There's the other way bacterial can regulate biochemical pathways, that's through feedback inhibition that we talked about in Chapter 8. But the more energy efficient way is to actually prevent the production of the enzyme in the first place. So the example that I'll show you is referred to as the Lac Operon model. The Lac Operon is just a set of genes involved in lactose degradation. And the effect is that the genes are only expressed when lactose is present and glucose is absent. Glucose is a preferred energy source if the bacteria can use these sugars. So glucose is going to be broken down first and lactose would be broken down second. So I can explain the Lac Operon using a bacterial growth curve. Now, in Lab 5, we put E. coli into different in media, and we use spectrophotometry. So this bacterial growth curve would be generated using spectrophotometry. We would have absorbance over time. So you take a sample periodically and measure the absorbance. And assuming we put E. coli in a tube with glucose and lactose, what's going to happen is we would get a preliminary bacterial growth curve, lag phase, log phase, beginning of a stationary phase. But what's happening here is the nutrients, glucose, is depleting. So glucose basically runs out, and this is where we would have the Lac Operon switch on. Okay, so remember the Lac Operon, those genes are only activated when glucose is absent. So glucose has to be gone, but lactose has to be present. So what would happen then is we would get a secondary growth curve where lactose is used. Here, glucose is going to be used. Okay, so the reason why cells do this is because it would be a waste of energy to produce enzymes to break down lactose when glucose is present. When the cells sense that the nutrient glucose is gone, it will activate that Lac Operon. So again, it's conserving energy by only producing one set of enzymes at a time. The cell can get plenty of energy, make plenty of ATP using glucose, so it's going to prefer to do that. So it's going to regulate its gene expression and not make the lactose degrading genes if glucose is present. The other topic with regulating gene expression is quorum sensing. And this is when cells sense the density, so the number of cells within their own population. So bacteria are able to communicate using chemical words. And so a cell will secrete certain chemicals, and other cells can absorb those, and basically they know they're in company. And the overall effect of this is that the population will only activate genes that are beneficial when they're expressed in a large mass of cells. Okay, so the cooperative activities in which biofilms are formed are controlled by quorum sensing, and bacteria can actually hold off, quote unquote, attacking the body until suitable numbers are present to go against the immune system. So just imagine yourself as a pathogen, and you have 100 of yourself, clones, trying to attack a body of 100 trillion cells. 100 of you are not going to basically be very successful. So what you need to do is recruit members, and bacteria recruit members by acquiring nutrients and reproducing. So when they sense the density is high enough, then they could activate genes that would benefit them, maybe produce a toxin to allow them to further invade the body. Now, on e-learning website, there is a video that I posted, and you probably have to download it. It's not a very large video. It's only a couple minutes, but it gives you an example of some of the ways scientists are applying bacterial communication and potentially blocking that communication as a new type of antibiotic. So this is going to conclude the Part 1, Chapter 9 video.